Welcome to this explainer. Imagine we're stepping right into a massive, cinematic computational landscape where we get to decode the literal machinery of life. Today, we're diving deep into the world of homology modeling. It's basically this relentless, data-driven engine that takes flat strings of amino acids and transforms them into fully realized, three-dimensional protein structures. Now, the entire foundation of this process rests on one really profound evolutionary truth. A protein's 3D structure is evolutionarily way more conserved than its underlying amino acid sequence. What that means is, even if sequences have drifted apart over millions of years, the overall 3D fold, it usually stays perfectly intact. And that gives us our core mantra, the real engine of our assembly line here. Unknown sequence, known template, new structure.
Step 1. Template search. Okay, let's dive right into this. We take our target sequence and rapidly scan massive structural databases, like the Protein Databank, using heavy-hitting algorithms like BLAST to find a matching relative. But, not all templates are created equal. Let's break down these identity thresholds. If we hit a template with over 50% sequence identity, we're golden. That's the highly reliable zone, where you can expect a very accurate model, maybe just 1 angstrom of root mean square deviation. Now, if you drop to that 30 to 50% range, it's still usable, but errors are going to start creeping into those flexible regions. But if we drop below 30%, oh boy, we enter what's literally called the unpredictable twilight zone. That is where serious errors and total fold mispredictions become a very real danger.
Step 2, sequence alignment. Make no mistake, this is absolutely the most critical phase of the entire operation. What's really interesting about this slide is how we visually map that query sequence directly against our chosen template. We're matching up those conserved residues and super carefully managing the gaps, the ones representing evolutionary insertions or deletions. The accuracy of this alignment literally dictates the fate of the entire model. Keep this in mind. A homology model is only ever as good as its underlying sequence alignment. Moving right along to step 3, model building. Now it's time for computational engines like Modeler or Swiss Model to take over the heavy lifting for the actual 3D spatial assembly. These engines follow a methodical, rigid body assembly process. First up, they copy the backbone coordinates right over from the template. Second, they predict sidechain placements using statistical rotamer libraries to basically hunt down the most likely atomic angles. And third, they modeled the loops to fill in those unaligned gaps. I've got to warn you though, loop modeling is uniquely tough because solvent-exposed loops are so incredibly flexible and almost always lack a clear template. Predicting their exact conformation is just a massive computational challenge.
And this brilliantly illustrates the exact moment a previously completely unknown target sequence actually adopts its projected 3D architecture. It's guided entirely by that alignment we just talked about. The structure is built, but hey, we aren't quite done yet. That brings us to step 4, refinement and validation. This is where we shift gears from pure construction into hardcore analytical precision. We literally fire off a whole battery of validation metrics here. First, we apply energy minimization to resolve any steric clashes. We're basically fixing impossible atomic overlaps like ring punches, which is just when a bond accidentally passes straight through a carbon ring. Definitely don't want that. Then, we run Ramatangen plots to make absolutely sure our bond angles are stereochemically legal. Finally, we calculate statistical scores, things like dope and process z-scores, just to ensure the overall fold actually reflects a realistic biological environment. And finally, real-world applications. Let's talk about why this whole computational assembly line actually matters when you're standing in a physical lab. So the crucial point is that homology modeling provides a totally essential shortcut for structural biology. You see, when proteins stubbornly refuse to crystallize for X-ray analysis, which happens a lot, this computational pipeline steps right in. It gives researchers the precise 3D structures they desperately need to run virtual screenings, map out potential binding sites, and ultimately design highly targeted drugs. It's an absolute game changer. At the end of the day, the whole shebang comes back to this relentless 4-step loop: template, align, build, validate. It's a continuous, beautiful cycle of computational discovery. And as our genomic databases absolutely explode with new sequences every single day, it really leaves you wondering, how many hidden cures and massive structural secrets are waiting out there right now, just waiting to be unlocked by this exact process?